Hello, biologists. Welcome to lecture. Today we're going to talk about biodiversity. What is biodiversity? Biodiversity is who lives there, how many species live there, what kind of abundance do they have, where are they found, and how do they interact? So that is biodiversity. We can measure biodiversity in a number of different ways. We can number the species, that would be species diversity, the different type of ecosystems, how organisms interact, their functional roles in those ecosystems. That would be ecosystem diversity and functional diversity. And some people get really excited about the different types of chemicals stored and used in each ecosystem. That would be chemical diversity. The type that we use as ecologists is species diversity, and more often, we're using genetic diversity. So genetic diversity is the variations in DNA of your species in a population. So population is one species in one place at one time. How do they vary based on their code, their A's and their T's and their C's and their G's? And we're using it more often because that variation in your genetic code leads to morphological variation, which may lead to different types of success. Here I have an image. This is all the same type of butterfly, but if you look from butterfly to butterfly, their sizes vary, their shapes vary, their color patterns vary, the hues vary. So all that different genetic diversity that we get from sexual reproduction and from mutation is going to lead to differences. And those differences can possibly lead to adaptations. And those adaptations can possibly lead to your species changing over time. And so that's why we're really interested in genetic diversity, is we think it's kind of the root of change and sustainability for a species as an ecosystem changes, giving us like the driver of natural selection. There are a couple different ways that people are tracking genetic biodiversity. One, there's a global genome initiative, which is trying to get scientists from all over the globe to collect genes from their species that they're studying and to store it. And how do we store it? We store it in things called biorepositories. So a repository is somewhere where you save something, bio telling you that you're saving something living. What do these look like? They look like a lot of different things. One is these cryogenetic freezing chambers. And in these cryogenetic freezing chambers, they're going to have little vials that are filled with DNA. They've taken the DNA out of the cell, they've amplified it to get a good copy of it, and then they freeze it. So we can freeze that DNA. The Global Genome Biodiversity Network has hundreds of thousands of samples total, which is really impressive. And they just store them into these freezing containers. It's really neat. We also can store information on the cloud. So instead of freezing a genetic piece of information, we can write down the sequence of A, T, C's, and G's, and then we can submit that to a database. One of the largest is the National Institute of Health's Sequence Read Archive. And this has so much data. Go ahead and read how much data on this slide here. An incredible amount of data. And that data is sitting there in the cloud, fully accessible. So anyone can go look at the gene sequences from researchers in other countries. So we're sharing it to get a copy of it so we know what exists where and how their DNA within species is variable. Super cool. And then the new cutting edge science of this, I've got a bunch of headlines from recent news reports for you. Memory crystals. So a memory crystal is a tiny glass square, and they write code on it. And now you have a physical storage unit for a massive amount of data so that you don't have to store it in servers or in the cloud. Because we may not use it for years and years and years. So it's like back in the day, we used to use floppy disks. Now we're going to store things on memory crystals. If you're interested in this technology, please look it up. It is super cool, but it's also cutting edge. So this is a field I think you're going to hear more and more about over the next several years. Really cool. One of my favorite ways to store genetic information is in a gene bank, which means you take a piece of the thing, either skin or muscle or an embryo or gametes, and you preserve it however that thing can be preserved. So some things can be dehydrated, some things will be frozen, some things will be preserved in a fixative like formalin or formaldehyde, and you just keep it. So you have a piece of its tissue containing its cells and containing its genes. One of the most sustainable ways to do this is seed banks. A seed is an embryo of a plant, which means it has all of the genetic information it needs to grow into a new plant. And a seed is a overwintering storage device on its own. So you just take your seeds and you can put them in little containers like being shown in this picture and slide them into shelves, and you have a genetic storage seed bank. There's really amazing seed banks in the world. One of the most famous is the Doomsday Vault. It is so cool. I'm really excited for you to see it. And when we're talking about seeds, we have to mention one of my favorite scientists, Dr. Vandana Shiva. She is incredible, and she is a huge advocate for seeds, and specifically this idea called seed sovereignty, which means freedom of oppression from seeds and collecting seeds and having access to agriculture and nutritious foods. Her nickname is the Gandhi of Grain. Her activism is inspirational, and she is a really incredible author. She's got like over 10 books. I can't wait for you to watch this little interview of her because she is wonderful. Excited for you. All right. So, if we're interested in biodiversity, we also have to figure out what exists on Earth. So, we're actively trying to name and describe and categorize what we know. We've estimated that we've done 1.5-ish eukaryotes so far, and we think that that's about 20% of the eukaryotes that exist on Earth. So we have 80% of the eukaryotes left undescribed, unwritten up. We know for fungi that we've only got 5% classified. 95% is left to be classified, and that is super exciting. There's so much work to do. If we go at the rate that we're currently going at, we think it will take us about 500 years to classify everything on Earth. How do we come up with those estimates? Well, we take articles that exist listing the species described so far versus the predicted species, and then we compare everyone's predictions and we come up with an average number of predictions. When we're thinking about biodiversity, we also want to think about where to look. So here I have a map of amphibian biodiversity. Take a moment. What kind of trends do you see? Pause the video if you want to. I see a very clear trend where here in this orange region, where we have the most number of species with these warmer colors, it's right along the equator. So the equator has a high diversity of amphibians. Amphibians, we know, need to have moist skin because they do cutaneous breathing. So we're going to find amphibians in places where there's liquid water. We can see this trend here when we go up to the areas where there's less liquid water, like the Arctic where it's frozen, or the Sahara where it's just exceedingly dry, parts of Australia where there's just desert. In these regions, there's not liquid water, and thus there's not nearly as many amphibians. If we look at biodiversity in general, we find a similar trend. Biodiversity does not exist equally distributed across the globe. In fact, there's small locations that have a huge number of species. We call these biodiversity hotspots. We estimate that 2.3% of the Earth's surface has 42% of terrestrial vertebrates and 50% of plants. That all of that in 2.3 of the Earth's surface. So what does this mean for us as ecologists? This means that we need to be focusing our attention, not blanket everywhere, but where we want to find the organisms that we're describing. So if we care about tracking biodiversity, the majority of our efforts need to be in these places that are highlighted in red and yellow and purple, because this is where we're going to find most of the species and where we need to be doing most of our research. So it's not an equal distribution. In fact, if we track number of species and area, we find this curved line. So if we have habitat area in square meters, 0 through 70, number of species from 0 to 35, there's a trend. You have a definite increase in the number of species as you go up, so up until like 10 meters squared, but then it levels off. And the difference in number of species between a habitat that's, say, 20 meters square and 70 meters square isn't as drastic as between 8 meters square and 0 meters square. So take a moment, look at this graph, and ask yourself, what is the independent variable of this experiment? What is the dependent variable? And what conclusions can you draw? Pause the video if needed. The independent variable, that is the thing that you change. So in this case, it is the measurable habitat area. The dependent variable, that's the thing that you measure. That is the number of species. So by convention, the independent variable goes on the x-axis and the dependent variable goes on the y-axis. What conclusions can you draw? The number of species in a habitat area increases very drastically up until about 10 meters squared. After 10 meters squared, the number of species still increases as the habitat gets higher, bigger, but it levels off. So the increase is not linear to the size of the space. And this goes with our idea of biodiversity hotspots. We had 2.3% of the Earth's crust having more than 50% of the plants. That's wild. So high area does not always mean high species number. When we think about marking our biodiversity and tracking our biodiversity, we also think about losing biodiversity. So we've talked about mass extinctions a couple of times previously, but I want to show you this graph again. Here we have extinction occurrence, 0 through 50%, and then we have millions of years before present time, and we have 5 purple dots. Each of these purple dots is a mass extinction event. A mass extinction is an extinction that goes well above the background extinction level. Because note, none of these ever drop down to 0, right? We don't have no extinctions ever. There's usually an extinction based on habitat change or natural disaster or just random events. But these are major, major extinction events. Each of these extinction events is going to give us a different period of time. So we see the background color changing as an extinction event occurs. And in these periods of time, after an extinction event, there opens up a huge new niche for everything else to start working in. And so after an extinction, we usually have diversification, and we have rapid evolution, and we have change, and we have growth. And so the loss of a species may be generally a net gain of a species, and it's definitely not the end of biodiversity, even if for a million years or a couple million years afterwards, the number of species on the planet is really small, it bounces back. And so we have this cyclical pattern of high loss and then high gain. All right, so ecosystem diversity is not just measured by the number of species, but that is an important part. The number of species is called the species richness, but we also want to look at the species composition. So who are the species in our habitat? How do they work with each other? What is the genetic diversity within that habitat? How are they distributed? All of the things that go into a functional population. When we're measuring biodiversity, we can use a species diversity index number. That's going to be the total number of species divided by the total number of individuals in a community of all species. When we think about diversity in an ecosystem, we have this idea that higher diversity leads to higher stability. It's called the diversity stability theory. Note that it's a theory. That means that it's pretty well supported. We've done a lot of experiments on it. We have good data supporting this. So as your ecosystem has more and more species, we have a bigger community. And as our community has more organisms that allow them to fit into their ecosystem in different ways, the ecosystem becomes more resilient. What does that mean? So here I've got a picture of moss, and then I have a porcini, which is the mushroom, and then I have a salamander on top of the porcini. So this is a good view of a resilient ecosystem because it's got a lot of different organisms in it, right? So we have the plant. I can see in the background there's vascular plants. I can see these leaves. So all of that material is going to get rotted by detritivores. Those are going to release the nutrients back into the ecosystem so there could be more growth. That fungi right there, it is a mycorrhizal fungi, so it's associated with all the plants around it, meaning that is swapping nutrients with plants and sharing different carbohydrates. That amphibian tells me that this is a moist habitat. If it's a moist habitat, there has to be food for the amphibians and a water source nearby for it to lay its eggs in. And all of these roles that we just discussed, they're not only one species that do it. So that gives us more than one species with a different role, leading to a more resilient habitat. We want more than one species with these roles so that we can say that they're functionally redundant. That means that there's more than one decomposer, there's more than one photosynthetic organism. The more species that you have, the more likely that your ecosystem will be able to bounce back from stress or disaster and will be able to support itself. So if you only had one decomposer and the decomposer died off for some reason, that ecosystem would be in big trouble. But if you have more than one decomposer, they're functionally redundant, then you can have variations in who is being the most efficient decomposer given the situation that your environment is in. That leads us to the insurance hypothesis. The insurance hypothesis states that the greater diversity you have of a species, the more stable that your ecosystem will be, and the more able it will be to withstand change without collapsing. So the greater the number of species is like a buffer against loss of species. Fascinating stuff, right? I hope that you enjoyed that and email me anytime with questions. We will talk soon. Have a good day. Bye.